The A F Harvey Prize is a really prestigious prize operated by the IET.
My nominator is the Dean of Engineering and he's from the UK here, and so for him the Harvey Engineering Prize has always been the holy grail. It's what he recognises as being the most highly sought after and the most prestigious of engineering prizes internationally.
I'm very thankful with the funding that comes with the A F Harvey Prize because it gave us additional resources to continue our work and to explore new, exciting research directions.
Over the past 2 decades, my team developed technology to stimulate the spinal cord, which allows us to restore walking after paralysis. And today, thanks to the prize, we have the opportunity to use the same technology to aim at restoring upper limb movement in people with tetraplegia. So I would like to thank you wholeheartedly to provide us with the opportunity to achieve this next breakthrough.
With the support of IET Harvey Research Prize, we will take this research further and particularly try to understand how we could apply smart materials to have dynamic control of those antenna performance.
I obviously do feel extremely proud to have won the award, both for myself but particularly for my research team. I think it's fantastic to have our work recognised and gives us the opportunity to take it forward.
Good afternoon. I'm Gopi Katragada, President of the Institution of Engineering and Technology. Welcome to this year's Harvey Prize lecture. It's going to be on harnessing deep learning and quantum technologies for antimicrobial resistance. This lecture will be delivered by our incredible winner of the 2023 A F Harvey Engineering Research Prize, Professor Rachel McKendry. You just saw a short film showcasing some of our remarkable past winners of the prize who have all been achieving great things since winning the funding. And as you might know, the prize, an impressive sum of £350,000, was set up in 2010 with a trust fund from the late Dr. Arthur Frank Harvey and is awarded annually to phenomenal researchers working in radar and microwave, lasers and optoelectronics, or medical engineering. Our search and selection panel looked globally for potential candidates, drawing on their expansive international networks to reward the most innovative workers in these fields. Professor Rachel McKendry is Professor of Biomedical Nanoscience and holds a joint position between the London Centre for Nanotechnology and the Division of Medicine, University College of London. She is also the director of the iSense EPSRC, IRC, a large interdisciplinary research collaboration in early warning sensing systems for infectious diseases. She is the 13th winner of the Harvey Prize. Today, we'll hear as she delivers her insightful lecture, and afterwards you'll have the chance to ask her some questions. Professor McKendry's research lies at the cutting edge of quantum technologies, deep learning and telecommunications for infectious diseases and public health. Her recent breakthroughs span from ultra-sensitive quantum nanodiamond diagnostics, this is for virus detection, all the way to mHealth and deep learning models for rapid testing in partnership with the Africa Health Research Institute in South Africa. Congratulations, Professor McKendry, on this very well-deserved achievement. I'll be back with Professor McKendry after the lecture, so get thinking of any questions and pop them in the chat. We will try and answer as many as we can. Cheers.
Hello everybody, my name is Professor Rachel McKendry and it's a huge honour to receive the Institution of Engineering and Technology's A F Harvey Prize 2023. The title of my talk today is Going Viral, Harnessing Deep Learning and Quantum Materials for Early Disease Diagnosis. I'd like to begin though by paying tribute to Dr. Arthur Frank Harvey. Born in 1910 and he died in 2006, Dr. Harvey's prize is his legacy to the furtherance of scientific research into the fields of medical, microwaves, radar or laser engineering. Dr. Harvey was said to be the most brilliant student the department has seen for many years, and also exceptionally brilliant and likely to go far, from the University of College of South Wales, Cardiff, Electrical Engineering Department. So it's in this same spirit that I would like to dedicate this prize today to my students who are also exceptional. Here is my group today, 4 PhD students and 4 postdocs, as well as my support staff and brilliant alumni listed on the right-hand side. And I'd also like to dedicate it to my wider team of collaborators, the iSense programme that you'll be hearing more about later today. This team is extraordinarily interdisciplinary, and it's very much in the spirit of my own career, where I've worked across the fields of chemistry, engineering, medicine, and now nanotechnology. And I have a joint position at UCL between the Division of Medicine and the London Centre for Nanotechnology. And this ability to work across different disciplines has shaped my research agenda. And it's very much around harnessing the power of new emerging nano and quantum sensors, as well as digital health, to address some of the greatest challenges facing society in biomedicine and global health. And really, I've been working at these interfaces for much of my scientific career. From the very early days, I became really interested in infectious diseases. Of course, we've just lived through a pandemic, but this slide, which I've taken from Anthony Fauci, highlights the diseases that over the past decades have been emerging and today are re-emerging. In red, you can see newly emerging infections. In blue, re-emerging and surging infections. And in black, deliberately emerging infections. And that refers to the anthrax bioterrorism. But it's clear, looking ahead and with the impact of climate change, infectious diseases will continue to emerge by their very nature. And our best strategy is to detect them early with a rapid response. The challenge of antimicrobial resistance risks our ability to honour the UN Sustainable Development Goals. Antimicrobial resistance strikes hardest on the poor. The treatment of resistant infections, for example, is more expensive. Untreatable infections in animals threaten sustainable food production for growing, hungry populations. Antimicrobials are fundamental components of all health systems. They're important for caesarean sections, hip operations, and all cancer treatments, for example. Antibiotics are today contaminating multiple water sources, and so risking our ability to provide clean water and sanitation globally. Moreover, the cost of AMR is increasing, and it's predicted to reach $100 trillion by 2050, driving an extra 28 million people into poverty. So it's crucial that we balance access, but also conserve antimicrobials with innovation to contain resistance so that they're available for future generations. But my career really began in the field of chemistry, and during my PhD I moved to the emerging field of nanoscience. It was a very exciting time in research, and I've titled this slide Adventures in Nanoscience, and it's really a tribute to my second PhD supervisor, Chris Abel, who recently passed away. And it was his approach to science, together with my first supervisor, Trevor Raymond, which really laid the foundations for my research. So a nanometer is a tiny length scale. It's a billionth of a meter. And at the time, a new type of microscope called a scanning probe microscope, also known as an atomic force microscope, had been developed. Now, it works much like an old-fashioned record player, where you have the arm of the record with a sharp needle at the end that traces over the grooves of a record. But in this case, everything had been miniaturized, and so we can begin to probe the nano world with a very sharp tip. And during my PhD, we were imaging different materials and single strands of DNA, but it was really this paper where we began to explore one of the most important but subtle interactions in nature, chiral discrimination, that I really became intrigued with the concept of sensing. Here you can see the tip of an atomic force microscope was tailored with a layer of chemical receptors, and the surface was patterned with two enantiomers. These are left and right-handed forms of a molecule. Essentially, it's like a glove on the tip trying to discriminate between two hands on the surface, and it's a nanoscale version of Louis Pasteur's famous experiment. And that was a really exciting time in our research. But simultaneously, I took up rowing and moved from being a relative novice to having a place in Cambridge University's Women's Boat Club, rowing for Blondie. And this experience really taught me so much about teamwork. Every single person in that boat, from the cox to the stroke to those in the middle, the powerhouse, I was one of those, to the girls in the back, played a key role. And it was vital that we all worked together, in our case to be Oxford, which we did. But this spirit of teamwork has percolated through all of my research over many years. But an opportunity came after my PhD to move to IBM's research laboratories in Zurich. It's a beautiful laboratory situated on the edge of Lake Zurich. And a few years earlier, they had published this paper showing that they could manipulate atoms. These are individual xenon atoms, which spelt out the logo IBM. And a few years earlier, they had received the Nobel Prize for the scanning tunneling microscope. So it was a huge honor to work at IBM. I was in the group of Christoph Gerber, who was pioneering the use of cantilever sensors to detect biological reactions. Now, instead of using the tip of the microscope that I explained earlier, this was about using the arm of the record player. We call these cantilevers. They're like tiny little diving boards. And here you can see arrays of 8 of them. And my research with Christoph involved functionalizing arrays of these with different capture layers, in our case oligonucleotides, and injecting different targets into solution. Now, when we saw sequence-specific hybridization between the target in solution and the probe on the surface, the cantilever bent, and we could read this out optically. And it was a fascinating sensing mechanism that translated a molecular recognition event into nanomechanical motion. So when I returned to the UK and had the chance to set up my own research team, I began to explore this in much more detail, exploring very basic chemical interactions, but also looking at increasingly more complex interactions. And this is when I first really began to look at antibiotics and the problem of resistance. The molecule that you can see on the right is a really beautiful molecule. It's also a very powerful antibiotic called vancomycin. In fact, it's one of the last line of antibiotics that's effective against some bacterial infections. Now, the chemist in me looks at the molecule and thinks it's incredibly beautiful. And I'm always reminded of an analogy by a professor in Cambridge, Dudley Williams, who likened that structure to a baseball glove. It targets a bacterial cell wall peptide, a mucopeptide, and the peptide sequence is shown here, lysine, dealanine, dealanine. And the way the two come together is like a baseball nestling in the palm of that baseball glove. And it forms 5 hydrogen bonds that you can see here. But resistance, which is increasing, occurs due to a deceptively simple mechanism, whereby this group is mutated to a lactate. It deletes a single hydrogen bond from the binding pocket, but it effectively renders the therapeutic ineffective, useless. And we began to look at this on our cantilevers, as you can see here, tailoring one side of the cantilever with layers that mimicked the susceptible bacteria and those that mimicked resistance. And in a series of papers published in Nature Nanotechnology and Nature Materials, we studied these interactions at the nanoscale. One of the most important breakthroughs is shown here, though, where we were able to look at the mechanism of a new antibiotic. It's called erytavancin. The molecule is on the right-hand side, and you can see all of the black sections are identical to vancomycin, but there are two slightly different new residues. They actually appear on the knuckles of the baseball glove by analogy, and so there was a paradox in the scientific community. Why was this antibiotic so good when the binding site hadn't changed? So with my team, we functionalized the cantilevers with lysine, dealanine, denalanine, the susceptible peptide, the resistant peptide, and a control. Now, if you look at the bending signals over time, when they're in a buffer solution, all three cantilevers behave in an identical way. But upon injection of vancomycin, we can see the susceptible cantilever bending, just as we expected. But upon a buffer washing step and then erytavancin was injected, we could see both cantilevers bending. And this is indicative that the new antibiotic was really binding to resistant bacteria. And to cut a long story short, we showed that the antibiotic is 4 orders of magnitude more active than vancomycin against the most difficult drug-resistant phenotypes. And this was a really important breakthrough. And more than that, we showed that it had a really interesting mechanism of action. It teamed up into molecular dimers, back-to-back dimers that could bind to 2 peptides simultaneously, and that's why it was so powerful. And this antibiotic has now been approved and is used in clinics, so it was really powerful to be able to make a contribution to a new antibiotic. But it was really after I returned from my maternity leave that a very interesting opportunity arose. A chance meeting with a company who were developing new sorts of mechanical sensors unlocked a whole new field of research for me. The picture that you can see here is of surface acoustic wave sensors developed by the company OJ Bio. What was really interesting about these sensors is they are actually found and made for smartphones, so they're found in smartphones. And we began to explore how could you begin to use those sensors in smartphones for diagnostics. So what was nice about these sensors is they don't require an optical readout. You could simply drop the sample on the sensor. But it was during the feedback from reviewers, when we were lucky enough to receive funding for this work, that there was a lot of questioning about why we needed a smartphone, that the data wasn't important. And that really for me was quite a light bulb moment, and it led me to bring together this extraordinary team, which is iSense. And I also had the opportunity to work with some of the very best scientists in the field of HIV, Robin Weiss and Dean Pillay. And so this question about what do we do with the data from diagnostics changed my research direction. And I set about putting together a program that brought together lots of traditional data sets, but also very unconventional data sets, from rapid tests, but also web searches and social media. Now, this was 10 years ago, way before the pandemic. We were thinking about using phones, using the battery power, the processing power, the screens, but importantly, the ability to send results in real time and with geospatial information to begin to understand how outbreaks are occurring and how we can prevent them. And it was at the beginning of the mobile revolution. I remember in one of my first talks talking about a supercomputer in your pocket. And of course, many of you will be watching my talk today on your phones. And it was a really timely point in the digital revolution. If you look at this graph here now with the handset shipments versus the date, you could see when we first began, which was around 2011, we were at the peak of the 2G phone era. But over the years, we've seen the transitions to 3G, 4G, 5G, and a 6G era coming now, with phones getting increasingly more common and powerful. So The group that I brought together is called iSense. It's an EPSRC-funded interdisciplinary research collaboration which aimed to build early warning sensing systems for infectious diseases. And for the past 10 years, it's been my absolute honour to be the director of this programme. It brings together researchers from many different universities with many different skill sets. And we've structured it around 3 key flagships: track, test, and treat. And you'll be hearing more about this, but it was about developing new tools for outbreak response, provider-directed care, but also strategically self-testing and treatment, which was a really new and interesting area. We applied this to flu, HIV, antimicrobial resistance, but also emerging threats like Ebola and COVID. And from the very beginning, we were interested in trying to pick up diseases at the earliest stage. Now, if you look at this typical timeline, following exposure to an infection, there will be an incubation period. It could be a few days, it could be much longer. Following onset of symptoms like a cough, a sore throat, a loss of smell, there will then begin to be health-seeking behaviours. We might go online and search our symptoms, or tell our friends, or phone the NHS. If it's really bad, you'll try to get an appointment with your doctor or turn up at A&E. And at this stage, a test may be taken and then sent to a laboratory to receive your result. Results can come back quickly, but for some forms of testing, it may take days or weeks to culture a bacteria. So there ends up being a very long time between exposure and a confirmed diagnosis. And during this time, a person may unknowingly pass on the infection. So we were interested to try to turn back the clock and pick up infections at that first onset of symptoms and develop tests that could work in decentralised settings, so outside of the laboratory, perhaps in a doctor's surgery, perhaps in home. But it became very clear that we needed to look at tracking outbreaks at a population, improving testing in the community, but also quickly linking people to treatment. And that became the paradigm of our programme. And here is our team. It's an extraordinary interdisciplinary team. The people that are shown here on the right are the management team. I like to think of them as in the same way I did about team spirit when I rode for Cambridge, where every single one of us has a role to play in the centre. And these are people with a background in medicine, in epidemiology, in public health, in engineering, computer science, geography, biochemistry, chemistry. Every single one of those people plays a key role in iSense. And we set about trying to look at what the user needs were and co-creating technologies, bringing together teams to look at new biomarkers, advanced materials, sensors, analytics, apps and dashboards, and then look to pilot and evaluate our technologies in the settings for which they were intended, both in the UK, but also looking at infections in low and middle income countries. We recognise the challenge of working across interdisciplinary borders. So we brought together an education alliance, and this is headed up by Neil Keegan, which takes and brings together the broad knowledge base of all our researchers, including postdocs, but also affiliated students. We also created exploratory projects, a flexible fund that allowed us to be agile and adapt to new opportunities over the years, and postdocs could lead these proposals. We created a partnership resource fund also, which allowed us to create mobility fellowships. So those early career researchers could go and be embedded within the World Health Organization and big public health institutions worldwide. And we had so much fun working with schools, working with the Science Museum, engaging with charities, running national competitions such as the app competition, and it created a really exciting ecosystem. And over the years, we've been looking to grow a network of excellence across the world. And I'll just take this opportunity to highlight some of our key postdocs who've gone on to leading roles. Noah Fongwen was a postdoc in London School and now heads up diagnostic access at Africa CDC across the whole of the continent. Polina Brangle was an engineer who worked in Molly's team and then in my group, who moved on to the World Health Organization and played a key role in COVID serology during the pandemic. Isabel Bennett was a PhD student in my group and now works for the Cabinet Office and had a career in the UK Health Security Agency. And Dunia Cherkaoui is now at the Foundation for Innovative New Diagnostics in Geneva. So in my talk today, I'm going to give you an overview of some of the highlights of our research. I can't cover everything, but I'm going to pick on some key examples to share with you the work that we've done. Under the TRACK programme, we've been looking at tracking outbreaks based on symptoms reported online at a population level. So not at an individual level, but looking at aggregated data. And we started with a focus on flu. Now, flu ranks at the very top of the Cabinet Office's risk register for civil emergencies, and both seasonal flu and pandemic flu is a major concern. But when we started, most of the disease surveillance focused on counting cases in hospital. It was also looking at the number of deaths from flu, and there is a really excellent, in the UK, Sentinel surveillance programme run by the Royal College of GPs across community cases across the UK. But the missing insight was what was happening in the community. So the bottom of the pyramid. People often like it to a submerged iceberg. Many cases can go just under the radar. And so we became interested in, could we use new and emerging data sources to begin to study flu in the community? And this is where we began exploring how symptoms reported online could be early indicators of a potential flu outbreak. And some of them you can see here. And to cut a long story short, and this is really work led by Ingemar Cox, Bill Lampos and Richard Pebbody, we were able to build early warning systems for flu and COVID, which have now been adopted in weekly government surveillance reports. The data has all been shared with Public Health England and the algorithms, and this is a really fine example of how early stage research can be used for public good. You don't have to take just my word for it. Here is a quote from the UK Health Security Agency's lead COVID-19 surveillance cell. This iSense system provided an essential surveillance system for the early indication of changes in COVID-19 activity. This was particularly notable at the start of the pandemic when the COVID Google search engine surveillance gave us one of the earliest indicators that the national lockdown was successfully reducing COVID-19 activity. And it built on years of work looking at flu, for tracking symptoms reported online in web searches and on social media, aggregated across large areas like all of England, for example, and providing an early indication by up to 1 week compared to traditional surveillance. We also used it to look at the effectiveness of live attenuated flu vaccines as part of a pilot study, looking at outbreaks at mass gatherings like pilgrimages to the Hajj, looking at digital risk markers, as well as the impact of climate change, for example, looking at heat waves. But really key to this work is the need for better data sharing. And we and others championed this call to the tech sector to encourage companies to work with researchers and government during the pandemic to find ways to share their data in a legal, proportionate, ethical and privacy-preserving manner, highlighting that this was vital in the public health response to COVID-19 and reflecting that the tech sector has benefited massively from public investment, and it's really critical that tech companies invest in society and share their data. And so we believe in future that tech companies really need to be partners in future pandemic responses. So over the course of the pandemic, we've been tracking what has been happening across the world and different groups using digital technologies in the public health response to COVID, from looking at better surveillance systems to diagnostics to monitoring the effectiveness of interventions and for public communications. And we identified huge opportunities, but also key challenges, including implementation and duplication. There were more than 38 symptom tracking apps in COVID just in the UK. Data access and quality is really key. Also, there is a need to publish the effectiveness of interventions, and regulators around the world were racing to catch up with the innovations that were happening in this space. It's very clear that there are legal, ethical and privacy concerns about bringing together these large data sets, and these frameworks need to be in place for future pandemics, as well as addressing inequalities and the digital divide. There are huge inequalities in access to digital technologies, both here in the UK and of course in lower middle income countries. And we believe there's a real need for public health, organisational and workforce leadership. It's very important to note that the WHO published its first report on using digital technologies for public health in 2019, so just before the pandemic. Within our test flagship, I'm going to move on to this now, we began looking at how could we use smartphones to improve diagnostic testing in decentralised settings. And when we first began this work, there were no self-tests commonly available. But we funded this project with John Edmonds, who is from London School. It was a joint collaboration between Flu Survey and iSense to explore this opportunity for self-swabbing. So working with their internet-based cohort, we sent out packages to people with instructions teaching them how to take a swab from their nose. Now this will be very familiar to many of us now. This is Claire demonstrating it, but I'll just point out that this was 10 years ago. We were piloting and evaluating this, recognizing that swabbing at home would be key to future pandemic preparedness. In this flagship, we've published a whole range of papers, and I'd really like to celebrate and give credit to all the teams that have worked developing new and interesting nanomaterials and new methods to diagnose disease, from our collaborators at Imperial, Molly's team, and also the team at Newcastle and the University of Strathclyde. But today I'm going to focus on a couple of key examples. The first, as I've mentioned earlier, is about tracking and exploiting different sensors that we can find in all of our smartphones. And one of them is the camera, which has of course evolved as more people want to take selfies and capture pictures of the world. And in this chart here that you can see, Joby Budd from my team has been tracking smartphone adoption worldwide in red and in sub-Saharan Africa in blue as a function of clock frequency power, and also looking at the number of megapixels in cameras from high and low end phones. And what you can really see is that smartphone adoption, as we know, is exploding. It's lower in sub-Saharan Africa, but it's certainly catching up. And the convergence in performance between high-end and low-end cameras is aligning. Joby, I would also highlight as being a really extraordinary engineer who spent much of the pandemic working for the UK Health Security Agency. Also began bringing together different data sources to see where we could use mobile phone connected tests. And this is a map of Uganda where Joby's overlaid network range coverage, as well as each of these dots is a clinic across the country. And he overlaid population data on top of this. And what you can see is large parts of the country have no network coverage, shown in grey. There's 4G coverage around the big cities. But we've been looking at targeting where mHealth technologies could be used, for example, in Uganda, in areas where there's still good network range, but where populations have to travel more than 5 kilometres to a healthcare facility. And Joby's been working with Africa CDC to help inform how these sorts of technologies could be rolled out worldwide. But it was really our collaboration with the Africa Health Research Institute, I feel, that's been one of the highlights of iSense, and working in partnership with them to understand how these technologies could be used within a resource-limited setting has been one of the highlights of my career. And this map highlights the challenge. It's from Our World in Data, and it shows that sub-Saharan Africa has the highest share of people aged between 15 and 49 who are infected with HIV. In the setting where we worked, and this work started in 2017 under a programme called M-Africa, funded by the Medical Research Council in partnership with iSense, there was 45% HIV prevalence in antenatal clinics. There's 9.5% HIV incidence for girls who are aged 15 to 19. 30% of people are unaware of their status, and only 50% diagnosed reach care within 1 year of a diagnosis. So a huge burden of illness on a community. And this, South Africa is considered to be, a relatively resource-rich country across Africa, but this part of KwaZulu-Natal is one of the poorest. But this graph that you can see on the right-hand side, I think, shows the extraordinary power of interventions. This is not work that we can take credit for. It's led by the Africa Health Research Institute, which is a world-class research centre in Africa. And what you can see is adult life expectancy as a function of time over the years. And in 2004, RE introduced antiretroviral treatments. And what you can see here is that life expectancy climbed by 11 years in the time before we started our intervention. So diagnosis is key, but the real game-changing breakthroughs are when people are linked to care and virally suppressed. So understanding the context, the environment, working together to co-create technologies was really so central to our collaboration with RE, and it was truly a collaboration among partners. And here you can see in this short video the work and the collaboration and the vision that we had. We worked with community health workers, nurses and peer navigators, looking at how we could look at integrating new digital approaches, including image capture systems and data dashboards, very much with a focus on capacity development and trying to meet their needs and understand where mobile interventions could be most impactful. The work has been published in Nature Medicine, and I think it's a beautiful example of how we can harness new innovations in digital technologies for global health challenges. And we set out by looking at how can we use deep learning methods to support HIV-based rapid testing. Now, the lateral flow test looked similar to the COVID test that we have all used during the pandemic, but if you look closely, these two different test types, test type A and B, are slightly different. One is slightly rounded, one is more rectangular, but if you crop around what's called the region of interest, the tests look very similar. And the challenge was to see if we could build systems to be able to classify tests as positive or negative. So a single line for negative and 2 lines for positive. But at that time, there were no real image libraries, and this was seen to be really cutting edge. The vision, and it's very much a long-term vision, was to build a data capture reporting system. You can see in blue on the left the current standard of care. A field work would go to someone's house, conduct a test, and interpret it by eye. We all remember how subjective interpreting a lateral flow strip was, particularly if the line was very faint. But in orange you can see our vision, which was to build an image capture system where the field worker takes a photograph of the test, automatically classifies the image, the result is confirmed to the participant and uploaded into a secure health database. With the advantage of there being no data loss, linkage to laboratory information systems, stock supply management, staff training, quality assurance of the tests, but really importantly, linkage to care and linkage into wider surveillance systems. And it was just such a pleasure to work with Cobus, Mariam, Dean and Val on this project and the wider group, including Karina Herbst. So this is the context for how we were conducting the study. This is one of our brilliant field workers capturing an image of the test together with Val Turbe, who now works with the Clinton Foundation. You can see the setting. It's extremely rural, under a tree near a homestead where there may be no running water or electricity. And in fact, the mHealth tray where the test are sitting proved to be very popular among the field workers because there's no table either. When we cropped around the regions of interest, here you can see the library. This is just a snapshot of the over 11,000 images that we acquired by 60 field workers using a Samsung tablet. And It was shown to be highly acceptable and feasible to both the staff and the participant, with really high consent rates of 96%. But what you can see is each one of those tests looks quite different. Some are slightly bluish, some are more reddish, some are in the shade, some are in the bright sunlight. And this is what a real-world image library looks like. And this was the first of its kind pre-COVID. And just before lockdown, we were able to conduct a very small-scale study comparing the ability of people, in this case nurses and community health workers in RE, to interpret the rapid tests compared to our machine learning algorithms. And what you can see here for test type A, there was pretty good agreement, 100% between the two nurses, and between 80 to 90% agreement between the community health workers who'd been trained for a year. On the right-hand side, though, you see quite a different picture with test type B. The agreement between the nurses has dropped slightly, but we can see that between the community health workers, the agreement varies from 61% to 94%. So just that subjective interpretation of test results left a lot of room for error. But with our deep learning system, which we call Classify, we showed that we could reduce the number of false positives from 11 to 0 and false negatives from 4 to 2. So cutting the number of false negatives by 50%. And you can see that overall this led to a huge improvement in specificity of 11% and a 2.5% improvement in sensitivity. Now, this was a small-scale study, but we are really excited about the results. And given that there are over 100 million HIV tests performed annually, and many more for other diseases, even a small improvement in quality assurance could impact the lives of millions of people by reducing the risk of false positives and false negatives. We made the algorithms all open source. It featured on the front cover of Nature Medicine. And during the pandemic, the REACT study in the UK used the algorithms and we worked together on 500,000 lateral flow strips, which is part of the world's largest serosurveillance study. And again, our algorithms, developed in collaboration with South Africa, showed really high levels of performance on COVID lateral flow tests. And we're now looking to work across a range of different disease areas and lateral flow strips, including on cholera, and are beginning a new collaboration with US regulators, the FDA. And I'm really delighted to show you Elliot Rogers, a new PhD student, who'll be leading on this work. Now, during the pandemic, lateral flow strips have really come to the fore. They've reached further and further than any other diagnostic test format. This very simple format doesn't require pumps or microfluidics and can be read by eye, has been adopted by over 101 countries worldwide. And this review that we put together with colleagues from around the world highlights some of the lessons learned from COVID. We can see on this timeline that whereas previously it took perhaps decades to get a new lateral flow test approved, during the pandemic this was dramatically condensed and regulatory approval came much more quickly. And we have been looking to see, can we engineer better lateral flow tests to target future epidemics and future diseases? Now, if you were to take apart your lateral flow strip, remove the cover, you would uncover some quite interesting engineering challenges. In there, there's cellulose, glass fibre, nitrocellulose, and the little red test line that you saw is created by 40 nanometer gold particles. When gold particles reach a certain size, they appear red due to their plasmonic properties. And you can see that we have the sample pad, the conjugate pad, the test line, and the backing card. But there's also a wicking pad and other components that go with a lateral flow strip. You can engineer them to detect an antibody, an antigen, or also increasingly detecting molecular targets like RNA and DNA. And it's a really exciting new field is to begin to integrate these molecular capabilities with the simple lateral flow test. If you look at the image on the right-hand side, though, you can see from a chemistry point of view that binding occurs due to the interplay of a whole series of chemical reactions which each have a binding affinity, an on rate and an off rate. And this is also really important in making better lateral flow strips. So we work together, this is work done by Jenny Brooks from my team, who is a really excellent physicist, to try to develop the most high affinity antibodies against one of the earliest markers of HIV. It's called P24 and it forms the capsid protein. And working together with Eleanor Gray and teams at QVQ, we developed one of the highest affinity antibodies for P24, combining in vitro structural, in silico and functional studies. This antibody was shown to have broad efficacy against all of the 50 global HIV subtypes and has a very strong binding constant. And we were able to collaborate with Molly's group and use these in a lateral flow strip to detect HIV with new levels of sensitivity that were unprecedented in the low picogram per mil range. This approach uses nanozymes, so inorganic catalysts, involving a two-step reaction. And I'd like to give credit to Molly's team here. And this technology is now being spun out into a company and is really exciting. But my team started to explore a new type of material. They're called nanodiamonds. Now, that might sound expensive, like something that we would find on Bond Street, but these type of diamonds have imperfections, and they're also grown under high pressure, high temperature conditions and micro milled. So they're actually very low cost. These, for the price per gram, we pay the same amount for these diamonds as we do for the gold particles in our assay. But they have actually really extraordinary properties, which we're very excited about and could unlock a new generation of ultra-sensitive tests. So the work was published in Nature. I'd like to give credit to the key people who were involved in this work. It's a collaboration between my team and John Morton's team at the London Centre for Nanotechnology. And we've taken advantage of this extraordinary property of nanodiamonds with precise imperfections to detect very low levels of virus. I'd like to give special credit to Ben Miller, who was a PhD student who began much of this work, and he has his own lectureship now at UCL. So I'm now going to look at a video which Nature has produced about our work, and we're just going to look at this now.
Diamonds are delicate enough to dazzle in jewellery and tough enough to withstand heavy industrial use. But lately it's their precise quantum properties that have unlocked a host of new applications. A pure diamond is made of a repeating crystal of carbon atoms, but diamonds really shine when they have impurities in them, and physicists are particularly interested in one impurity. That's when a carbon atom is replaced with a nitrogen and a neighbouring carbon atom is missing. Together, the nitrogen and the vacancy act like a single entity, an NV center. This center has a physical property called spin. The NV spin is like two tiny compass needles. They either both point up, giving a spin of 1, both down, spin -1, or when one is up and one is down, spin 0. And it's this spin that makes NV centers so handy to physicists. If you shine a green laser on an NV center, it re-emits light at a lower red frequency. But an NV center emits more light when it's in the 0 spin state than when its needles are both pointing up or down. And researchers can use this to check what the spin is up to. A microwave at just the right frequency can flick an NV center from 0 spin to up or down, a change that causes the red light to dim. Scientists can use this for all sorts of applications, like diagnosing disease. Tiny diamonds can be attached to a particular virus molecule. The researchers use microwaves to brighten and dim the light the diamonds give off. This helps the diamonds stand out from the crowd of light pollution in a biological sample. Spotting a diamond means that the virus is present and the disease is detected.
So let's take a deeper look at why nanodiamonds are so interesting. Now, if we look at the energy level diagram shown here, we can see that negatively charged NV centers in diamond have electron spin triplets in both the ground and optically excited states. So if we use wavelength that's at 550 nanometers, we can optically excite electrons from the ground to the excited states, conserving spin. For spin ms equals 0, the electron decays back to the ground state, emitting a red photon. For spin ms equals plus or minus 1 states, the electron can also, though, alternatively decay via this metastable singlet dark state shown on the right. And in this case, no visible photon is emitted via this pathway. The reduction in fluorescence is equal to the probability of decay via this singlet start state. And it's typically 5% for high density NVs, but up to 30% in single NVs. And we can see this really clearly now in what's called an optically detected magnetic resonance plot on the right-hand side. So as you saw in the videos, if we apply microwaves at just the right frequency corresponding to this gap in energy, we can pump electrons to spin populations and cause and modulate the intensity of the fluorescence. So what you can see on the right-hand side, if we sweep the frequency, there's a significant dip at just the right frequency of 2.87 gigahertz. And this corresponds, as I say, to this energy difference, delta E. Now, if we thereby sort of put a resonator and apply a microwave field just below the surface of the lateral flow strip and apply a modulating microwave field, we can effectively cause the nanodiamonds on the strip to modulate. So what you can see here is a flashing light, and that's important because the nitrocellulose lateral flow strip is also autofluorescent, but being able to modulate the light enables us to pick out the diamonds on the test strip against the background. And the analogy I like to think about is a lighthouse flashing periodically. And so we can begin to see tiny things on a surface and become really quite extraordinarily sensitive, working in the frequency domain. In this case, the modulation is at 4 Hertz. We also spent a lot of time focusing on optimizing the nanodiamond capture chemistry, looking at non-specific binding, but also developing chemistries to tailor these to functionalize them with capture antibodies and molecules that we were interested to target, looking at evidence that we'd functionalize them, tracking things like their size through each stage of the functionalization. But I'd like to really focus now on this important study, which really tested the limits of detection of this system using biotin-avidin, which is a model system, one of the highest affinity interactions in nature. What you can see on the right-hand side is as we titrated the nanodiamond concentration, from the first image, you can clearly see a test line at the highest concentration, but as you look at lower concentrations, the test line becomes much harder to pick out. But if you look at the modulating signal below, we can clearly see the signal even at very low concentrations. And on the right-hand side, you can see a plot which shows we can detect approximately 10 to the 6 gold particles, which are traditionally used in these tests. But with the nanodiamonds, we can improve this sensitivity, detecting only 1,000 particles. And with the modulation, we're down to detecting just 26 particles on a lateral flow strip. So there's an extraordinary 10 to the 5-fold improvement in sensitivity. More than that, we can use it for really interesting biological samples. And in this work, we optimized the assay to detect a key biomarker of HIV using isothermal amplification, a method known as RPA, and we're able to detect it on the lateral flow strips, detecting single copy sensitivity with a 10-minute amplification step, so a very short amplification step, and evaluated both negative plasma and standard clinical samples from UCL Hospital. So we were able to detect HIV up to a week earlier than current tests. Now, I'll say that this research is still very much at an early stage. We use this microscope to read out the fluorescence, but we're working towards seeing if we can develop a point of care test and exploring new ways to modulate the signal and read it out. And I feel this could be such an exciting new field of research for infections, but also more broadly. And a whole range of applications are emerging, from diagnostics to looking for viruses in the environment. And my team of researchers are looking and exploring these new research directions. And I think there will be much more to come in this area of exploring new quantum sensors. But now I'll pivot just to the last flagship. We looked at track, test, and now we're going to focus on treat, because following a diagnostic test, the key thing is to get people onto treatment quickly. And during this program of research within iSense, we've developed a whole series of online care pathways being led by groups at UCL, building on work they'd done previously, but also creating versions in isiZulu, the local dialect for use in South Africa, and also data dashboards to make sense of large data sets as part of this treatment as prevention trial. In future, as we look ahead, we think we need a generalized format for online care pathways and clinical user journeys in a variety of settings. So wherever someone is testing, if they test negative, the results will be still reported into surveillance systems, but they can get quick access where needed to post-exposure prophylaxis, or prompted for repeat testing and given health promotion advice. Whereas for a positive test, this could trigger a confirmatory test and linkage to care, or where appropriate for certain diseases, quarantine and contact tracing. And I'm really delighted to say our work has now been featured in the International Pandemic Preparedness 100 Days Mission. The Diagnostics Implementation Report was just published, and two key parts of iSense research were featured in there. So to conclude, I think it's been a hugely exciting research journey working across the interfaces of nano and quantum technologies and digital technologies to address some of these huge challenges facing society, from global health to the discovery of new antibiotics. And the research I've told you today has highlighted some of these opportunities and the work that we've done, which has at least in part contributed to the discovery of new antibiotics, disease diagnostics, and early warning sensing systems that have been adopted and used during the pandemic. The field of quantum sensors, I think, is just in its infancy and will be emerging over the next few years and could transform how we currently diagnose diseases. I'll also highlight that co-creation with end users and working in multidisciplinary teams has been really key to all of our research, and there is huge potential for benefits which are both to health but also economic benefits to society, to patients, populations, health systems and governments. And as I've alluded to earlier, the impact is not just on infections but also on non-communicable diseases. And I'd like to finish also by coming back to the A F Harvey Prize 2023 and to say a huge thank you for the funding, which we are so grateful for, and it will help us to explore new avenues of research, particularly in the area of developing rapid tests for antimicrobial resistance. And it will link also to a new hub that we've just had funded, the Digital Health Hub for Antimicrobial Resistance. And I'm also really excited to begin a new public engagement activity celebrating the hidden heroes of diagnostics. So looking at the engineers and physical scientists behind many of the diagnostic tests that we use today. So finally, a thank you to all of my collaborators, my funders, my family, and to UCL for enabling me to conduct such interesting work over many years. Thank you.
Thank you everyone for joining in, and Rachel, what an excellent presentation. I thought I was going to be listening to words of wisdom on biology, but then you had sensors in there, you had materials in there, and you threw in the diamonds as well. I really enjoyed the presentation, and I'm sure all the listeners did as well. So the way I look at your presentation, for my thinking at least, is that when we think about innovation in these times, it's usually at an intersection of technologies. And in your talk, you very clearly showed the intersection of material science, computer science and biological science. Maybe I start off with a question there, Rachel. Which of these areas, materials, computers, or biological sciences, do you think is your area of expertise, or are you good at all these areas?
Thank you so much, Gopi. And maybe I can just start by saying a huge thank you to IET for this award, which I actually have with me at the moment. So I'm really, really delighted to receive this, not just for myself, but for my whole team and collaborators. It really is really such an honour to receive this award. To your question about working across disciplines, I think that's been the hallmark of my career. As I sort of showed that my background is in chemistry, I moved to working in engineering and then in medicine. So that's been hugely exciting for me. I would say I'm an expert in some ways, in some aspects of each of these, but it's really the breadth of expertise, I think, which is unique for me. And I also work with really outstanding subject matter experts. But what's really driven me in my career is the ability to tackle the biggest challenges in society, and that's very much involves working at the interface of new materials, data science, medicine and biology.
Yeah, and there's a question from the audience as well on sensors. Where was the bottleneck, if at all, or where were the challenges? Was it in the sensors or in the data analysis? And this is a question from Kevin Tibbetts.
Oh, thank you, Kevin, for the great question. And there are absolutely many significant challenges that lie ahead. So much research is needed and development from many of the engineers that are watching today. With our nanodiamond work, which we're really excited about, some of the biggest challenges are around sensitivity and specificity, but there are also translation, scale-up, manufacturing challenges, and also challenges getting new sensors adopted and implemented within health systems. So if we focus on the sensitivity challenges, many groups have been looking at a whole plethora of particles and ways to improve the sensitivity of those tests. We were really excited about the use of nanodiamonds, and bigger particles certainly help us to gain sensitivity, but there are also challenges picking up tiny signals in a complex background. We took advantage of this ability to modulate the spins of the nanodiamond NV centers, which was a unique approach. But non-specific binding is equally a challenge, where particles stick to surfaces non-specifically. And so that necessitates working with chemists and biochemists and material scientists to reduce some of those background signals. We talked a lot about sensitivity, but specificity is equally important. So designing controls and really good control experiments is really key. And in work we've just finished recently, we've evaluated a whole range of different respiratory viruses to look at specific and sensitive diagnosis. And as I mentioned, we're at the beginning of our journey to develop the next generation of quantum enhanced tests. So there's a long road ahead to take a good idea that was published in Nature through to a scalable manufacturable test that can be used in resource limited settings. But we're really excited and making good progress on that journey.
Thanks, Rachel. So nanodiamonds, what makes them special? For example, there has been a lot of work done in carbon nanotubes or carbon nanoparticles. What is the closest alternative to nanodiamonds, if you would? And what is it about nanodiamonds that makes them stand apart? Because at the end of the day, they are carbon still, right?
Yeah, so many groups around the world are looking at new materials. So some are working on, as you say, carbon nanotubes or graphene sensors. within our work, we made a discovery that using nano diamonds, but with a very precise imperfection, an NV center. So it's where a carbon is replaced with a nitrogen. It creates a vacancy center adjacent to it. But it's this NV center with a negative charge that gives diamond a really interesting optical property. It's very bright, it's relatively low cost, but it is this ability to selectively modulate the emission, which I think is quite unique. There are other NV centers where that can work, but we really were excited to use this property for the first time in an in vitro diagnostic application. And the reason why that is important, so lots of people look at materials that might be brighter or more fluorescent, but the background membrane, the paper membrane in the strip, is also fluorescent. Yeah. So it's quite hard to see a fluorescent particle on a fluorescent background. But by modulating the signal, turning it on and off, or modulating the intensity of the signal, we could lock into that change. I think on the video I gave the analogy of a lighthouse. It's really hard to see a light across an ocean, but once it starts modulating periodically, you can lock onto that signal. And that allowed us to separate the periodic signal from the background, and we could achieve this really quite unexpectedly good improvement in sensitivity, 100,000-fold improvement in sensitivity. The other aspect that I really like about diamond is it is carbon chemistry, so it's amenable to all sorts of linkers, so that we can tailor and orient receptors on the surface of diamond to make it bind specifically and sensitively. It's relatively low cost. In our lateral flow strips that we make in my lab, it's perhaps the lowest cost of all the components that go into it. So yeah, it's got many interesting properties which we're looking to explore. I should say, since we recorded the video, we've been very fortunate to just have been awarded a major programme from the UK government and the EPSRC and then NIHR to create a national centre to look at harnessing a whole range of quantum sensors for healthcare applications. So we'll be looking at new sensors beyond, but including nanodiamonds.
So congratulations on the funding, Rachel, and again, well deserved. You mentioned, of course, the lateral flow strips, and of course the fact that they are cost effective, and I believe that's what the images that you showed as well were of, where you were able to use AI and get maybe better results than manual interpretation. The question from Ray Bouchard is, did you find Android or iPhones equally capable for this research and reporting?
Thank you for the good question. It's a great question. So in that study in South Africa, we used Samsung tablets that were already being used by the field workers. So it was just one type of test, one type of device. But we're actually just beginning a large project with one of the main international regulators to begin looking at Android versus iPhone. And in fact, in my lab, we've looked at 10 different phones, both low end and high end. I can't yet say which one is best. It's not yet published, but they're both performing pretty well. The other thing I would say, building on the work we did in South Africa with the Samsung tablets, we made our algorithms available for a very large study during COVID called REACT. It was the world's largest sero surveillance study. And in this study, over 500,000 images of lateral flow tests for COVID in the UK were uploaded. And given that iPhones represent, almost half of the UK mobile phone users use an iPhone, and our algorithms performed very well on those, we think, they will perform equally, at least equally as well.
Yeah. So what's important, Rachel, it's of course, the camera of the phones which are taking the pictures. Is it the resolution? Is it the color sensitivity? Is it the dynamic range? Would you?
So we're beginning to look into all of those aspects. I would say one of the challenges, though, with phones in cameras is not always all the specifications are made available, and also with software updates and software corrections to phones, it's sometimes hard to disentangle those things. But that is the purpose of using of this new project with the regulators, to look at some of the opportunities and maybe pitfalls of using cameras in this way. But we all have our phones with us for most of the time, so taking advantage of the huge investments that have gone into making those and the cameras that are available, I think is a really interesting area of research.
Yeah. So also you've used AI, and today with the progress with LLMs and Gen AI, and also the recent release from OpenAI O1 version, which is apparently good for biology as well. Are you looking at these new developments in AI?
So we have a new digital health hub for antimicrobial resistance, which will begin to look at all of the big breakthroughs. I'm not currently looking at LLMs, but we are very interested in, and others in our in UCL are exploring them. I think they will have it will be a hugely interesting time looking forward, given now the developments that we're seeing in large language models. I even had applications for jobs in my groups from that have declared themselves to have been written by ChatGPT. So it is an interesting time, and many are fearful of these developments. But I feel like we need to evaluate them and look at them. We have to be mindful of the biases that might creep in in using them in health. But I'm optimistic, and I think it's an interesting area of research.
So since you mentioned bias, the data sets that you use, of course, you're working in the African continent, but how do you make sure that they're inclusive to be able to be applicable across the globe, or are you focused on the continent right now?
So the work in KwaZulu-Natal that was shown in the video involves working very closely with the Africa Health Research Institute. It's a Wellcome Trust funded demographic surveillance centre in the heart of KwaZulu-Natal. So really, they should I give them credit for developing the infrastructure to ensure that demographic biases and are included in their surveillance systems. We worked with them to support them move from a paper-based method of collecting data to a digital approach. And actually, we learned a lot from working together. It was very much a collaboration. There are biases in many different data sets, I would say. Even in the UK, there are biases in even data within the NHS based on people who may attend clinic where others don't. So we always have to be mindful of biases in different data sets. I'm particularly interested also in the use of online data, such as search or social media. And this is the work that's really led by Ingemar Cox in my team. Again, that has a bias based on people who go online and search, but it provides really quite an important, unique insight as to what is happening and doesn't rely necessarily on clinic attendance.
Thanks, Rachel. I'll end with asking you a question from the audience again. And this is a career question, and it's for all the young researchers for sure, but this is particularly for women. And this question is from Claire Mansfield. Do you have any career advice for women working in this field?
So we absolutely need more women in this field, Claire. So please be encouraged, pursue your dreams, be bold and confident in your career moves. I would say in my career, so I don't come from a background, my parents weren't in academia. I've often been, the only woman, the first woman to be promoted as a professor in my department. For the iSense program, I discovered fairly recently that I was the first woman to lead that program since the scheme began in 1988. That's really quite a long time ago. We absolutely need to bring more diversity, more women and more underrepresented groups into engineering and science. Please be confident. And to all the men out there and for everyone, we need to support women like Claire.
So Rachel, what I'll request is a couple more questions. I'll try to combine them, and also if you can give your closing remarks to the audience, it would be great. One of the questions is around treating symptoms versus actually a cure, and how your work might impact this area. The other question is maybe just a quick factual question on connections with ZOE health studies, so ZOE health studies. And so if you can answer those two and wrap up with any closing remarks that you might have, would be great. And on your closing remarks, it would be great if it is on what you're seeing as the future of wellness and health.
Thank you so much, Gobi. So I'll try and answer those questions to start with. I think the first one was on diagnosis and tracking symptoms versus the cure.
Exactly, symptoms versus curing the root cause.
So I'll highlight that my research has all been about trying to detect the very early stages of infections, where there may or there may not be symptoms, and obviously fast access to treatment can really lead to the big health outcomes. I can't quite see the question online, but there are many working on cures for diseases, which is incredible. My work is really on diagnosis, but faster access to treatment can then lead to complete recovery and better health outcomes, potentially saving lives. And the second question, can you just remind me?
Yeah, any relation of your work with
to the ZOE app?
Zoe.
Unfortunately, no, but I've been a great admirer of the ZOE work. But I'm happy to wrap up. I'm going to again thank IET and Arthur Frank Harvey for this incredible award. I really take it myself, but it's taken on, I hope I've given full credit to all of my team, all of my students. Looking to the future, I think as engineers, if we can contribute to improving universal healthcare access and coverage across the world, to focus on contributing to making a better world where healthcare is affordable and accessible, and that we leave no one behind, is an extremely important objective. And yes, thank you again to IET for this brilliant award. It's a huge boost to my group. I'm really grateful for the funding and looking forward to pursuing our research on the use of nanodiamonds to detect antimicrobial resistance, and also a public engagement activity which is looking at the hidden heroes of diagnostics, so celebrating the many engineers who have developed the diagnostics that of today and of the future. Thank you.
So Rachel, congratulations again, and I love your message of affordable and accessible healthcare with diamonds in tow. So congratulations, and I request the audience to stay on for a video on the future funds, which will be put out by IET right now. So please stay on and do watch the video on one of IET's activities around future funds. Thank you.
Gives you stills And this isn't the work of our teachers, this isn't any schoolwork, this isn't anything we're forced to do, it's things we actually want to do.
Some people like me, I come from a lower socio-economic background, so might not necessarily have those contacts with companies to get that work experience, to get that first step on the ladder, and things like this can actually get you onto that ladder so you feel like you can have a fulfilling career.